Hello, fellow Group 11 members. I hope that you are all doing well. My name is Richard, and welcome to this week's presentation. So let me go over the description for this week's topic again. Hemoglobin is a protein which binds oxygen in the lungs and releases it in the capillaries. Interestingly, your body controls this intake and drop-off of oxygen by having different pHs at the two locations. So our goals for this week are threefold. First, we need to go over how oxygen binds to heme. Next, we need to explain the binding capacity of oxygen to heme in the lungs and how that differs from that in the capillaries. And finally, we need to talk about how CO2 interacting with H2O can affect the pH of the capillaries. So let's start with the structure of hemoglobin first. So hemoglobin is a component of red blood cells, or RBCs for short, and it is a quaternary structure consisting of four subunits, two alpha-beta dimers. So as you can see over here, this, for example, would be alpha-1 beta-1, and this would be alpha-2 beta-2. So these dimers associate together to form the hemoglobin tetramer, tetra meaning four. And within each of these groups contains a heme group. So what is a heme group? As you can see in this image over here, this is what's known as a heme group, and each heme group contains an iron molecule, or Fe here, and it has a pyrrole ring over here, methyl group, and a vinyl group. So again, on this image over here, the heme is denoted as these blue discs surrounding each dimer. So hemoglobin can exist in either of the two forms, deoxyhemoglobin or oxyhemoglobin. So deoxyhemoglobin, also known as an oxygen-free form, is also known as the T or tent state, has less affinity for O2, and is in a non-planar configuration, while oxyhemoglobin has a bound oxygen molecule, is also known as the R or relaxed state, and it has more affinity for O2 and is in the planar configuration. So these two images over here better show the differences between deoxyhemoglobin and oxyhemoglobin. So in the case of deoxyhemoglobin, you can see that the iron atom is lowered from the heme group's plane without the binding of O2. So again, over here, it is not in the planar configuration, so there is no binding of O2 whatsoever. While oxyhemoglobin, the iron atom is flushed with the heme group's plane, so it's in the planar configuration, as you can see over here, and that does allow for binding of O2, as you can see over here, O2 binding. So how does oxygen bind to heme? So hemoglobin must be converted from the T state to the R state. So first, the iron ion moves into the plane of the porphyrin. Then the histidine residue that is bound in the fifth coordination and its associated alpha helix will also move to the plane of the porphyrin ring. The dimers will then rotate 15 degrees with respect to one another, as you can see over here, rotating 15 degrees. And by doing this, undergoing this rotation, it provides a pathway for communication between the subunits. So the presence of oxygen on one subunit is immediately communicated to the others so that they change from T to R. And that is how oxygen binds to heme. So let's now move on to the binding capacity of oxygen in the lungs versus the capillaries. So it is affected by both the pH and the partial pressure of oxygen, given in units of torr. So you can see on the right over here on this diagram, hemoglobin is the S curve, which denotes cooperative binding, or in blue, and myoglobin, seen in red here, is denoted for non-cooperative binding. So when O2 is transported in the blood from the lungs, it is done in conditions such that the partial pressure of oxygen is equal to 100 torr and the pH is equal to 7.4. This means that there's a high binding capacity of oxygen in the lungs, or in other words, O2 is harder to be released. When we move from the lungs to the tissues, there's a noticeable decrease in the partial pressure of oxygen as well as the pH, as seen here, 20 torr and 7.2 respectively. This means that there's a low binding capacity of oxygen in the capillaries, or in other words, O2 is released more easily. So deoxyhemoglobin is stabilized by ionic bonds or salt bridges. And notice in the high pH and high partial pressure setting, no salt bridge forms, so the T state is not favored. Meanwhile, in low pH and low partial pressure, salt bridge forms, so the T state is favored. So to better illustrate what's going on over here, you can see this image on the right-hand side of the deoxyhemoglobin. So again, the side chain of the beta-1 histidine 146 becomes protonated. Once that happens, the salt bridge with beta-1 aspartate 94 forms, as you can see over here. And what this does is it stabilizes the quaternary structure of the T state, and that leads to a greater release of oxygen at actively metabolizing tissues. So just to reiterate again, there's a high binding capacity of oxygen in the lungs versus a low binding capacity of oxygen in the capillaries. And finally, we need to discuss how pH is affected when carbon dioxide interacts with water. So the carbon dioxide in tissues diffuses into the red blood cells, as you can see over here. So CO2 reacts with H2O to form carbonic acid, H2CO3, and this carbonic acid dissociates to form HCO3 minus and this H plus. So when we have this H plus, it drops the pH inside, it drops the pH inside the red blood cells. So if there is no water present, then the carbon dioxide cannot react and form the carbonic acid, and because of this, no H plus forms, so the pH will rise instead of fall. And if we recall from earlier, an increase in pH will increase the affinity for oxygen, causing it to bind tighter. And because of this, the tissues will not be able to collect the oxygen, which can then lead to cell and tissue death. Thank you very much.